to be here tonight and for the opportunity to tell you about our nanoscale tools for deciphering biochemical signaling in the body through imaging. So the need that we're trying to address is to visualize the chemical content of the body. And so we feel that the language of the body is built with biochemical signals, whether you call them biomarkers or signaling in the brain and body. And I feel that we need to understand the identity of these biochemicals, their location in the brain and body, and the amplitude and timing of the signal. And so if you take the image on the left here, and think about it, if we take something like an MRI, what we're really gaining from this imaging modality is structural information or functional information. But yet we know that as you're listening to me and processing this and remembering it, what's really happening is a whole multitude of biochemical signatures. As one neuron in the brain communicates with the next one, it is this entire flux of biomarkers. It's sodium and potassium and calcium during what's called an action potential. And then one cell releases neurotransmitter to the next one to start that whole process again. And this happens really very quickly in the body. And we know that alterations in the signaling are really important for disease. But yet when we take that image of the body, right, we're losing all of that information. And so what we're trying to do as a lab is to combine nanosensor development and imaging modalities to visualize the chemical signatures in the body so that this image becomes rich with biochemical information. So currently, apart from my lab, what is done now is basically, I should take a step back. So our approach is to combine these novel probes for biochemical detection. And each one of our sensors or contrast agents or whatever you want to call them for illuminating the chemistry of the body is basically composed of 3 or 4 parts. The first one is a recognition unit. So we find a small molecule or an enzyme or protein that binds to our biomarker of interest. But because these recognition elements quite often don't have any sort of signal that we can monitor, right? So they could be bound to our biomarker or not, and we don't really have any way to address that. We combine them with a reporter that reports to us through some sort of imaging modality what's going on here with this biomarker of interest, whether it's bound or unbound. Because we, practically speaking, want to use these in the body, we need to join these 2 elements together, right? So they need to be in close proximity to one another. And if we just injected them into the body, they would diffuse away and they would never work together as a unit. So we combine these with a scaffold to hold them together. And then quite often we put a targeting moiety on this so that they stay in the region of interest in the body that we want to measure. We then combine these with advanced imaging technologies, and we really take a design approach to this and think about what sort of information do we need to get from this imaging technique? Do we need really good resolution, or do we need really good whole body imaging? And we kind of look at what we need to get out as an answer to our problem. And we use this iterative process to go back and forth and combine reporters with imaging modalities to really get to the heart of the question. And then finally, we characterize these in animal models to make sure that they work in animals the same way that they work in, a cuvette on the benchtop. And ultimately, our goal, although we're not there yet, is to be able to use these in humans as well, so that we can create an atlas of signaling in the human body. So why isn't this done now? So right now, the tools that are available, not considering these nanoscale probes, but if you were just using the tools that are available right now, what we've found is that chemical analysis in the body is a trade-off, like all measurement techniques, really. So we know that these biochemical signatures are very important because if you take a sample from the body, let's say a blood sample or a tissue sample, we have great instrumentation that can read very specifically the chemical that's involved in whatever process you're trying to measure. But it requires removal from the body. And not only, is this difficult, so I mean, a blood sample is easy, right? But if you want to take a tissue sample, then, that's a little bit harder. And really what you've given up is that time information, right? So if you take one sample, you don't know what happens 5 minutes later. There's no way to know unless you take sample after sample after sample. And it's just not practical when you're evaluating a patient, say. One could implant a probe and identify a specific chemical in the body, but now you've given up your spatial information because there's just that one probe in that one area. There's been some great progress in optogenetics, which are fluorescent proteins that can light up some of these signatures, but obviously those are best in laboratory scale animals. And finally, as I mentioned before, we have the image of the body, right? So something like an MRI, but we've now given up all of that great biochemical information we got from taking a sample, and instead just get this structural information. It's fantastic and non-invasive, but we've really given up that chemical information. So what we're trying to do is really bridge these two areas, option 1 and option 4. Can we bridge those so that we can get this great biochemical information through imaging the living body? So I'm going to tell you a story today about a particular area that we're focused on, but no matter what we are trying to measure in the body, there's certain design requirements that we really need to meet. When we design a new probe, we have to start really at the fundamentals, and we have to think about what probe characteristics are important. So first of all, obviously size is very important. It needs to be able to fit in a region of the body in which you want to make a measurement. We have to think carefully about the dynamic range and tune our sensors accordingly so that we're measuring in the correct dynamic range. Sensitivity is also very important and I think overlooked sometimes. And so quite often in the area of diagnostics, when we're thinking about, let's say COVID diagnostics or some sort of diagnostics, people quite often say it's like looking for a needle in a haystack, right? You want to look for a very small number in a sample. But really here, in our particular application, we're not looking for a needle in the haystack. Instead, we're looking to be able to measure a certain amount of change in our biomarker on a background of that biomarker. And so I like to refer to it as measuring the addition or subtraction of a few pieces of hay to that haystack, right? We need to make a very sensitive measurement on a very high background. And it's a very challenging problem. And then we also have to think about reversibility when we're designing our probes. We want to be able to have this real-time readout, this amplitude and timing. So there's going to be rises and falls in that level of biomarker in the body, and we need to be able to design a probe to address that. Once we have designed the probe and characterized it on the benchtop, we then need to think about how to get it to the area of interest in the body. So, I've mentioned the brain several times. And it's a very tricky region to measure in, right? So if you inject a probe into the body, how do you know it's going to get to the brain? We have to design that in there, and we have to figure out ways in order to direct it to the region of interest in the body. And we have to minimize the amount of background that we have from probe that is there, but not really in an area of interest to us. It might just be reporting background signal. We then need to think about imaging through tissue to minimize scatter. We need a quantitative signal to everything that we do. And then we validate these in animal models. And sometimes this means we need to go back to the drawing board. So once we go into an animal model and we find something that's a challenge we didn't think of, sometimes we need to go back and redesign our probe and then go through the entire process again. So the story I'm going to tell you today is about real-time chemical imaging, is imaging signaling events in the peripheral nervous system, and in particular acetylcholine analysis. And for this particular project, we needed to think about, what is the peripheral nervous system? This is the way that your brain communicates with, let's say, the organs of your body. We needed a way to measure acetylcholine, which is a neurotransmitter that is really important in this communication. And we needed to be able to measure it with very good spatial resolution and very fast. And we were willing to give up, and very sensitively, and we were really willing to give up invasiveness. So we were okay with invasive technique for this measurement. So basically, we relied on fluorescence for our sensor. So before I talk more about the science and the fundamentals of this, I wanted to mention the area that we were working in. And in particular, we were working with the NIH SPARC initiative. And this SPARC stands for Stimulating Peripheral Activity to Relieve Conditions. And so what it seeks to accelerate development of therapeutic devices that modulate electrical activity in nerves to improve organ function. So what is this? So as I mentioned, the brain is controlling the organs in your body. As you are listening to this, your heart is beating, and it is beating regularly, and you don't have to think about controlling that. This is all just happening in the background, and it happens correctly all the time. And it's not just a one-way communication either. Your brain is telling these organs to function, but there's also communication in the other direction as well. And so really what's happening is that these nerves from the peripheral nervous system are in contact with the organs. And as that nerve fires to control the organ, there's a small release of acetylcholine between the nerve and the organ. And that is the biomarker that we're trying to measure. This is indicating that there is proper communication between the nerves and that organ. And so the rest of the SPARC initiative is thinking about things like, can they control these organs with microelectrodes? So electrodes have gotten very, very sophisticated. There's really great ways to interface them with the body. So can they use those electrodes as a kind of therapeutic? So let's say you want to control bladder function or you want to control heart function. Can these electrodes be placed on nerves that communicate with these organs in order to act as a therapy and, better bladder control for those who suffer from bladder control issues or whatever organ of interest? And so where we come in, because we can measure biomarkers so specifically and so sensitively, is that direct readout. So what is a dose for a microelectrode that's placed on a nerve that's controlling an organ? It's not as straightforward as taking, let's say, an Advil or some sort of other, drug where the dosing profiles are so well worked out. And instead, they have to use those electrodes to control the nerve. And so what is the dose of an electrode? It's a little bit hard to know. But where we come in is that we can tell them exactly how much biomarker is released from that nerve so that they can very quickly tune their electrode to be releasing the amount of biomarker that they know will effectively communicate with the nerve. So it's kind of that instant readout of what's going on. So specifically, acetylcholine is the key biomarker. And if you think about the communication between one nerve and the next, one neural cell in the next, what we have is a small area called the synaptic cleft, and acetylcholine is released from one cell to the next in that communication. This also happens at the organ surface in which these nerves, actually it's called innervate the organs, and that release of acetylcholine tells that organ to function, whether that's the bladder or that's the heart. That's the communication pathway there. And so if we place sensors in this area, we should be able to give a real readout to what's happening if you stimulate this nerve here. So in this particular story, what's really going to be important for our probe characteristics is, really the size is going to be very important because we're working in a very tight area, and the sensitivity is going to be key. So our approach is to use DNA nanotechnology to create that scaffold. So if you remember, I said there's three important elements. There's the recognition element, there is the reporter, and there's the scaffold. And in this particular application, the scaffold was key because we had to have a very homogeneous, programmable scaffold. And DNA origami is a just beautiful example of what can be done by programming DNA to build structures. And so basically what we start with is a template of, half of the DNA. We can take little pieces of staple strands, and what you can do is put them into solution, heat it up very slowly, and then cool it down very slowly. And when you do that, these DNA basically find their complementary strand, and they can build structures. And there's free software online that you can use in order to predict that structure. It works really, really well. And then you can synthesize those structures and anneal them. And for us, this is a fantastic scaffold because it's completely programmable. We can put different functionalities, so we can add on our reporter molecules, we can add on our recognition elements, and we can do this at a very high yield, and it works very well every time, and you get structures that are very, very reproducible. So in particular, the way that we built these nanoscale probes, if you think about this fundamental structure, we have the recognition element, we have the reporter that the binding event happened, we have this scaffold, and in this case, we did add a targeting moiety. So at that synaptic cleft that I showed you earlier, as acetylcholine is released from one cell to another, there are these receptors, these acetylcholine receptors. And this is where all the action with the acetylcholine happens. So we want to target a very small percentage of these receptors that occur on the cell surface with our sensor. We have the DNA scaffold, and we've built kind of a small dendrimer shape with this, kind of a branch structure. And on this, we've placed our recognition element, our reporter, and a targeting molecule. So in this case, our recognition element is acetylcholine esterase, which is an enzyme that is found in the body, and its job is to degrade acetylcholine. When it does that, it produces an acid. So because this has no signal on its own, an enzyme can be, degrading acetylcholine all day and we would never know it, right? So we need to pair it with a reporter. And in this case, we used a fluorescent pH indicator. As the environment becomes more acidic because of the acetylcholine esterase, there's a localized change in pH, and our fluorescent reporter increases its intensity. This is a reversible process. As the acetylcholine goes down in the area, this goes down as well. So we can see a rise and fall in fluorescent signal due to the enzymatic reaction. And then finally, we add a targeting moiety here, BTX, and this targets this nicotinic receptor and binds the sensor in place so that we can make our measurement. Our nanosensor fabrication is from the bottom up. So basically, we synthesize these oligos, which is 1/2 of the DNA helix. And before we anneal or form the structure of our complex, we basically functionalize it with our fluorescent reporter. Once our fluorescent reporters are attached to the oligomers, we can then anneal the structure. So we heat it up very slowly, cool it down very slowly, and in solution, these spontaneously form. Once they have spontaneously formed, we can kind of clean up the solution, and we're left with these very nice structures that have our fluorescent reporters attached. And chemistry on the ends where there isn't fluorescent reporters so that we can do further functionalization of our scaffold. In particular, the next step is to attach our enzyme, acetylcholine esterase. We can't do this before we anneal the structure, so that heating up process would denature or basically ruin our acetylcholine esterase. So we do this after we anneal the structure. We can there are surface lysines on the enzyme that naturally occur. We can use a malamide linker and functionalize our enzyme and then add it to the thiol groups that we've placed on the end of our DNA and conjugate through a thioether bond. Separately, we can add the BTX to the sensor through a different set of click chemistry. And basically, we use a TCO linker and basically end up with a stable bond to our recognition element. So two different click chemistry reactions are used to attach two different proteins to very specific positions on this DNA scaffold. We then purify our sensor by size exclusion chromatography. So basically, size exclusion chromatography is we use these beads, and we can place our sample at the top. Big molecules come out first as we run solvent through here, and so we can basically purify our compounds by size. To get a purified sensor at the end with none of our reactants still in the solution. And then if we characterize this, we find that as we hoped with DNA, and the reason that we chose it, we get a very high yield of attachment of all of our different molecules. So this is an absorbance spectrum, so how much the light is absorbed by our final sensor. And we get different peaks from our DNA and protein, from our linker, from our fluorophore, and from the dye label that we placed on our targeting ligand, so that we knew it was there. And if we use Beer-Lambert's law, we find that for every one scaffold molecule, we designed 4 enzyme molecules that would be attached. Our yield is more closer to 3 enzyme molecules. We designed 4 fluorescent reporters, and we almost get 4 on every single scaffold. And we designed 2 targeting ligands, and we come close to 2 targeting ligands on every single sensor scaffold. So we think that we have a lower yield of our enzyme because of steric hindrance. So basically, once we have a couple conjugated to our structure, it's harder because there's not space for more enzyme molecules to come in at the same yield. So as I mentioned, once we go through and design and characterize our sensor, then we like to take it through its paces in an in vivo environment. And so we chose the submandibular ganglia for our proof of concept. So as I mentioned several times, the nerves are coming in and innervating the organ and releasing acetylcholine, and that's our signaling molecule. And this is really true for basically most of the organs of the body. And so we could have chosen any one of them, but we chose the submandibular ganglia. And this is basically the nerves that control the salivary duct in your mouth. So when those nerves fire and release acetylcholine, they're telling your saliva gland to release saliva. And we chose this because of the proximity to the outside surface, which just from a technical point of view for a proof of concept makes it much, much easier for us to work out a lot of the imaging parameters. So it's kind of a first step to a gateway to the other organs of the body as well. And I have to apologize, my little arrows all shifted somehow on this image here, but really this is an image of what the nerves look like. And the ganglia, so these green things here, are shown in these spots here. Let me see if I can get a better laser pointer. Okay, so better laser pointer here. You can see the ganglia are these little bumps here on each of these nerve fibers. And there's a lot of these ganglia. And so if we label with a fluorescent compound, these ganglia here, you can see them. There's many of them on these nerves. We can then place an electrode on the nerve. And what will happen is that we use the electrode to stimulate the nerve, label the ganglia, and watch the acetylcholine being released. And it's all just extremely tiny and difficult to work with. So I really have to give Junfei and Michelle a lot of credit for having worked so much of this out and having such, calm hands in this procedure. So our imaging, basically, we use just standard imaging, fluorescent imaging modalities. We place the mouse upside down on the microscope stage, and then we can use an objective, a microscope objective, to excite the fluorescence of our sensors and then collect the signal back using a light source for excitation. That fluorescent signal is then collected back and imaged on a very fast CMOS camera with a beam splitter so that we can image side by side both our signal and our internal standard, which I probably haven't mentioned much, but is located on our targeting ligand. And so the advantage of having an internal standard is, if you think about it, when you're trying to make a very sensitive measurement, if you have a very bright signal, there can be multiple reasons why you have that bright signal. You could be measuring a lot of what you're trying to measure, right? And then your signal gets really bright, and you hope that that's the case. But the other thing that could lead to a bright signal is that you could have just a lot of sensor in that area of interest. And so if you use an internal standard to mark, every single sensor, that signal doesn't change, and you can always know, okay, this how much sensor is there, and then you can make a really quantitative measurement on top of that with your reporter molecule. We have to surgically expose the submandibular ganglia, and basically you surgically expose it, and then using a very, very small spatula to basically pull the SMG up so that it's closer to the microscope objective for imaging. Because the microscope objective can't get down deep into tissue. So we use this little spatula to pull it up and keep it basically steady for imaging. And I mentioned that delivery is always a challenge. I think that in this case, the delivery of the sensors to the SMG was really a very major challenge of this project, and really took a lot of work to figure out how best to get the sensors to the SMG. We would have liked to have been able to just take some sensor and bathe it in the region of interest, label the SMG, and start making our measurements. But really, no matter how talented Michelle was or is with doing this, this procedure to take the SMG and stretch it out over the spatula for imaging, there's still just a little tissue here that is shielding that SMG. And so it would just take a very, very long time for our sensor to kind of diffuse through, make its way through that network of tissue to label the SMG. So instead, what we had to do was take a glass capillary and pull it into a very fine point. So this is glass needle. It's probably a few microns in diameter, and then take it at a 25-degree angle into this tissue here. So you can see this needle just a little bit here in this image. It's at a 25-degree angle to the spatula, and she brings it in here and inserts it into the SMG, and then just pumps in a few microliters of the sensor that are in solution into the SMG area. And you can see that image here. So this is the tissue delineated here. You can't see it here, but there would be a spatula behind here to pull it taut. And that needle, and she's pumping in little bits and little bits of the sensor in solution. And so she's pumping in more and more, you can see here. And every time she pumps in a little bit of sensor, more of it labels in the tissue in solution until finally, oh, she got a really good injection there. A lot of sensor labeled that tissue, and you can actually see the cell borders as that, and it labels very, very quickly. That targeting ligand binds to the receptors very, very quickly, and the excess sensor diffuses away. So we don't have a high background of sensor that doesn't belong where it's supposed to go. It diffuses away, and we're left with a nicely labeled ganglia here in this area of interest, and it's pulled tight so that we can do some good imaging here. After, this is just a nice picture of what it looks like after it's been labeled. This has been isolated from the animal and imaged on a very nice confocal microscope. The green spots are cells. This isn't our sensor here, but the pink spots are our sensor. So you can see how the pink spots show that around the area, around the surface of all of these cells, these ganglia cells, these nerve cells, there are our sensors waiting there to measure acetylcholine as it's being released. We then have to take it back through analytical characterization again. So this is an ex vivo calibration. What we've done is gone through that whole procedure that I just showed you. We pull the ganglia up, the SMG up on that spatula, we label it with sensor, and then we take that ganglia and then ex vivo, so outside of the animal, we can then place it under a harp, which keeps it steady and taut as well. And then that gives us the ability to add small amounts of acetylcholine ourselves at very well-defined concentrations. And we see how does that sensor respond when we've added increasing amounts of acetylcholine. And as you can see here from these images, the more acetylcholine that we add to the tissue, the brighter the signal, right, which we would expect. We can then analyze this data and build a concentration curve. So the signal that we measure is here on the y-axis. The concentration that we have added of acetylcholine is on the x-axis. And we can build a calibration curve. And once we've built that calibration curve, not only can we now know what our lower limit of detection is, how small amount of acetylcholine can we measure, in this case 228 nanomolar, which is quite small. So we can measure baseline levels of acetylcholine. But more importantly, once we make a real measurement that, from our whole experimental setup, what we can then do is say, okay, we've measured, let's say 200, and we can then pull this back on our calibration curve and find out exactly how much this signal change leads to what concentration. So that means we can make quantitative measurements in vivo as we move forward. So this is what it looks like. So this is the setup. We have injected our sensors into a mouse, into the SMG, and we have placed those electrodes on the nerve that's coming into the ganglia. This is an image of what it looks like. The cell is outlined with these dotted white dots. And we've basically taken a region of interest, which has quite a bit of sensor here next to the cell, because if you remember, the sensors label all around the cell. So we just chose one little region of interest and analyzed it. And when we analyze the change in stimulation here, so if you look at the blue line here, we get a baseline. At the arrow, we've basically turned on our electrode and stimulated the nerve. And we can see clearly that acetylcholine is released upon stimulation with that electrode. and then it goes down after the stimulation is over. The blue is the error bars. This is n equals 3, so 3 independent experiments. Our stimulation parameters for our electrode were 3 volts, 10 hertz, 10 millisecond duration, and 100 pulses in this particular case. The green line is interesting. It's our control, and so nothing happened. But what's interesting about it is that the control basically consists of the exact same sensor that I told you about. But in this particular case, we have basically not put on the recognition element. So that enzyme, acetylcholine esterase, that should degrade acetylcholine and leading to a change in pH, has been left off. So all we have left is the scaffold, the targeting ligand, and the pH sensitive fluorescent reporter. And so basically, when nothing happens, this is telling us that upon stimulating this the acetylcholine release, it's not leading to a change in pH that's happening biologically, right? So everything that we're measuring is because of the sensor so for acetylcholine, we are measuring acetylcholine basically, and that is what is happening in this biological environment. The dose dependence of this is important. If we think back to the problem that we're trying to solve, it's to produce a tool for the rest of the Spark community to use for their experiments. They want to know whether or not, when they've implanted an electrode, what dose to use. And so our sensor needs to be able to measure changes in that dose. And in this case, the dose is the electrode that we're using to stimulate the nerve. And so if we change the voltage of our electrode from 3 volts to 10 volts, we see a much greater change in acetylcholine. So basically, the more voltage we pump into the system, the more acetylcholine that's being released. If we change the number of pulses but leave the voltage the same, we see a similar sort of thing. So more pulses release more acetylcholine. And of course, this is completely reversible. So if we stimulate the nerve, we see acetylcholine release, go back down to baseline, we can repeat the stimulation and it happens again, and we go on and on and on. So this we feel could be an important tool for the rest of the community, because we can give that a dose-dependent release profile information in real time. And it's quantitative, which is also important. So if we take that data and our calibration curve that we produced ex vivo, we can choose a region of interest, look at what happens in that region of interest, and then go back to our calibration curve to find out just exactly how much. So we now know our y-axis, right? So, we see this fluorescence profile. We can use our y-axis to go back and say, okay, what is exactly our acetylcholine concentration that's being released? And in this case, with the data that you saw on the previous slide, it's 2.6 micromolar that is released upon stimulation. And then I'm, a big believer in imaging technologies, and this is the reason why. It's maybe expected, maybe unexpected result here, is that when we place this electrode here on the nerve that's coming into the SMG, I guess what I expected from this stimulation profile would be that across the tissue that defines one SMG, that acetylcholine would just be released kind of from everywhere from this nerve stimulation. But that's not what we saw at all. And so if you look at the raw image, this is one SMG that's being stimulated. And what's happening is that the stimulation is starting on one side of the tissue closest to where the electrode is and propagating across that tissue. And we use this software called Aqua that has been developed by Kira Pascanzer at UC San Francisco. That is a good tool for visualizing this propagation behavior in fluorescence imaging data sets. And it was a very nice tool for us to use even in the peripheral nervous system. And I think shows very nicely. This is the Aqua image. So basically, we've just processed that signal. And you can see very clearly that that signal, we'll wait for it to start over again, but you can see very clearly that that signal starts here. Okay, starts here at the top upon stimulation, and you can watch that propagation pattern occur across that tissue. And whether or not that's because where we had the electrode placed on the fiber, or whether that's because acetylcholine is, somehow, a signaling molecule it's in, propagating this, I don't know. I don't have the answer to that. Hopefully, it'll be a solved problem later, but it really highlights the importance of having this imaging technology, because if we had no ability to look at the spatial information, we would have missed that altogether. So in summary, we've built a new tool in order to visualize acetylcholine release in the peripheral nervous system. And we really feel that these DNA-based sensors are a great tool to just be able to, really homogeneously define sensor size and chemistry, so that we get the same sort of readout again and again. And what we're really working on now is expanding this to other organ systems, particularly imaging acetylcholine in the heart. We're pairing this with a new nanoscale probe that we've almost got finished now for norepinephrine, so that we can do 2 neurotransmitter imaging at once, which will be important in the heart. And we'll probably try to make this a little bit smaller yet, so that we can get target into even smaller spaces between cellular communication. So with that, I definitely want to thank you for your time. Take any questions that people have, but more importantly, I want to thank my fantastic lab. They're just such a great team who have a very interdisciplinary background and really work together to bring these different backgrounds to problem solving and trying to build new tools and new methods. And in particular, I'd like to thank collaborators, James Monahan and Kira Pascanzer, my postdoc, Donna Yang, who really has done so much of the synthetic work, Jun Fei, who started a lot of this, Michelle, and Nick, who have done so many of these imaging technologies. So with that, I'd be happy to take questions.
All right, fantastic. As expected, it was a great talk. So we will jump straight into the questions. And just for chat, if you have questions that you're formulating, please feel free to get them in. We got, there's not really a time limit on it, so we'll just go as long as you got questions. So the first question we have is, do the targeting moieties get taken up elsewhere, or are they specific enough to only go where you want them to?
Yeah, so they definitely only go where, well, it's maybe a little complicated. So we try to, by our delivery techniques, basically deliver them to a specific area of interest. Anything that does diffuse away after the labeling occurs is definitely going somewhere and probably labeling, but it would be a pretty small quantity. But the kind of, I don't know, maybe secondary question to that is, once we go to like different organ systems, we actually have to work out different targeting moieties, because each organ has different receptors that we want to be able to target.
Okay, that makes a lot of sense. Let's see. Is there structural deviation of the receptors after binding of the sensor?
So I don't think that there's structural deviation, but we have actually, once we've bound to that receptor, it's no longer able to then sense acetylcholine. And so we only bind to about 10 to 15% of the receptors that are there. And we feel like that probably isn't distorting the biology, but definitely once those receptors are bound, they're no longer being used as receptors in the body.
Okay. Let's see. So this is a question specifically about the annealing process. The chatter was wondering if you could talk a little bit more about the time scales and the temperature scales of the annealing process.
Yeah, it's very, very dependent on what we're trying to build. I think that this particular one is, I don't remember the exact temperature and profile, but it takes place over a few hours.
Okay.
And it goes up, I want to say about 60 degrees. So it's enough that we would damage the protein, but it's not hot. So it's somewhere in between.
Okay.
It's not a very complicated structure, so we have a little wiggle room.
Oh, okay. All right. Are there any issues with variation between animals in terms of trying to establish the reproducibility?
That is a fantastic question. So between rodents, no, it's been very reproducible.
Okay.
If we were working in other species, I don't have an answer to that yet. It should be similar.
Okay. Interesting.
Yep. Yep.
Is there a difference in the km of the enzyme? I guess that's acetylcholase, ACHE.
Yep. Yep. Yeah.
That's my lack of biology coming out right there.
Yeah. So when we do the if I can kind of read into the question a little bit, when we do the covalent attachment to the sensor, it is a little less active than the native acetylcholinesterase. We do change it a little bit for the worse when we do that covalent attachment.
Okay.
Yep.
All right. Rama, if you had a different idea there of what you were trying to ask, feel free to follow up. So this is you touched on this a little bit, so I guess some of this may be a little redundant, but is this a method that you think will be able to be applied both to other molecules and other parts of bodies, whether it's in a rodent sort of model system, or maybe potentially in people or something like that, or is the development of this technique something that would have to be repeated each time? For instance, for a different molecule like acetylcholine, or again, if you move from a rodent model into larger animals or people or something like that.
Yeah, so there's two parts to that question. The one is going and measuring a different molecule. So let's say norepinephrine. And so because we have a modular sensor, we should be able to just pop in a new recognition element and a new reporter and move on. And I always tell my students it will be very quick. In reality, in reality, they give me the eye, and I know it's not as quick as that, right? We always end up going through so much, redesigning every single time. But I think that if you think about its use in, let's say, rodent, and then if you worked in other animals, or hopefully humans someday, I think that would be the advantage to our particular sensor over things like optogenetic approaches, which have to be genetically modified into animals, which works really, really well in mice, but maybe a little harder to envision in moving up in species or even human. And I think that would be the advantage for us. This is just DNA, and it's just protein and a small amount of reporter. So it should be able to be something that's biocompatible and easy to use in different species.
Okay. So I take it from that answer that you ultimately, that it may not be, in the next 5 years, but somewhere down the line, you envision this being something that could be used as potentially as like a diagnostic technique in people or something like that.
I would love to see, this really have impact as the field moves forward for this electrical, therapeutics.
Mhm.
I think it could be a good tool to really individualize a treatment for a particular person, which I think will be important in that particular field.
Very cool. So I'll give any chance for any last questions to come in. But in the meantime, I will go ahead and say thank you to Dr. Clark. It's a fascinating talk. Uh, it's really cool to see sort of the edge of the leading edge of what work is being done. And it was it's very well explained too. For someone like me who is not biochemistry was a long time ago. I was able to follow it and understand what you're doing and why, and that's a really cool thing. So that speaks highly of your ability to, not only the technical work that you've done, but your ability to explain it to someone who's not overly familiar with it. Um,
Thank you. Thank
so,
you.
yeah, I think it looks like that's all the questions we got. So, I will say thank you to you. If I could get you to hang around for just 1 second,
Sure.
we'll talk briefly. For everybody else, I hope you will come back out next week. We'll be back again at same time, same place, and we have Abhishek Padar from University of Toledo speaking next week, who's a biologist. So, some more great work to look forward to there. In the meantime, have a good weekend. Everybody be safe. And if you're not vaccinated yet, go get your shot. And I hope to see you all back here next week. And have a wonderful one.